I'm Russ Reed, and I'm the PI of what we call Beta Skills. And Beta Skills stands for Biomedical Emerging Technology Applications. And this is a grant that we have from the NSF ATE division. We've been running this grant now for several years. Actually, this is the 6th year of the grant. And it has really transformed this grant because the way it was set up was to look at skill standards, then it became professional development. And so I'm really pleased today because part of professional development is an idea that we had amongst our subject matter experts to actually put people into industry settings so that they could see what it's like to actually be in industry, because it's kind of paradoxical that you could have an instructor who's teaching students about industry, and yet they may never have had an industry experience. And from my 20 years of doing this, traveling across the country, I can tell you that most instructors have not had an industry experience. So we're really excited because there are going to be multiple presentations today. Bear with us because some of these presentations are being made virtually for people who were here but who have returned back to their sites, and it ranges from the East Coast to California. So, and actually if we were to include Pratima actually flew in from India to do her fellowship. So we're very excited. It's quite a unique program. And the first person we're going to start with is Dr. Jeffrey Kane. Jeffrey, welcome.
Welcome. Thank you. Thank you, Russ. I have some slides here. So who am I? I'm Jeff Kane, and I came from the University of Massachusetts at Amherst. And I teach a one-year biotech program in applied molecular biotechnology. Most of my students come in to learn hands-on how to do techniques in the lab, do research, and then they go to small biopharma companies in the Boston area. I've worked at a couple of small companies before, but I really am lacking in production experience, really pharmaceutical production. I was fortunate to be placed at Pfizer in Sanford, which is a very large company. They produce drug substances and conjugates that end up in vaccines that you can get at your local Walgreens or CVS. What did we do during this time? Well, we did a lot of training when we first got there. We learned a lot of stuff that I was not familiar with. Everything at that facility from batch access to pest control. I mean, we had to learn the whole gamut. But the focus was on GMP and CGMP with aseptic technique, and we did so much gowning training and degowning training because everything has to be protected in that facility. My first real big learning experience was learning pharma speak. All these companies have their own language, and they're all these letters like IPA and PAL, and the weirdest one was WIFI. It's something that I've never talked about, but water for injection. What did we do on a day-to-day basis? We met with all these different departments, we met the leads, we met people working on the floor. We went to what they call MSAT, Tech Ops, and fermentation, purification, validation, all the way through the entire company in this time. And what I was doing there is I was trying to find out what each group actually has as a task and what they did that fits into the production of these materials. And everywhere I went, I asked, what are the skills that students need that I'm going to teach? What do I teach them so they can fit into your company? All right. And the biggest thing I got back is general knowledge of the field, very important. They need to know what they're doing and what it means. And they need to have attention to detail and integrity, because you're going to be signing off on something that's eventually going to go into a human body. What else? We went on all these tours. I mean, these were literally the places we went. We went in these places that are amazingly built, but everywhere we went, we had to gown in and do everything. We couldn't really touch anything, but we really were able to see exactly where that 2,500 liter fermenter is that's making the component that ends up in the Prevnar vaccine. And then we went all the way through what they call Train 1, Train 2, Train 3, and the gene therapy section. The gene therapy section is to me, a cutting edge that I really was interested in. Besides making the drug substances, they actually do make one drug product there that actually goes out. We didn't have to gown every time. There were occasional times where we went into QC and some other places where we just had to wear a lab coat. So my goal coming here was to learn more about gene therapy and be able to talk about monoclonal antibodies production so I can teach students so that they can get jobs again. A few things happened on the way. We were supposed to be here for 8 weeks. I had a late start because I had some personal issues. There were some holidays in there that kind of knocked things off, like the 4th of July. And then 1 week I broke a tooth, so I had to find a dentist. So there's always complications. The biggest thing at Sanford was their lead drug for gene therapy for muscular dystrophy actually failed third stage trial right when we got there. So they're changing what they're going to be producing there, coming up with some other things, and it was a little bit of a shuffling things around. So meetings that were scheduled had to be rescheduled. Some people couldn't meet at a certain time, so they came to the next week. So every day was a little different. What I did learn was a completely different point of view from working on research, teaching students hands-on how to solve this problem right in front of you. It's a different point of view when you're making a large amount of a particular drug for an entire society. Because they make stuff for the US, for Europe, for all over the world. There are different aspects that we talked about, different ways of looking at things, like thinking about it as tech ops and manufacturing science and technology. These are people that are working towards the same thing, and every time they make thousands of liters of this product, it has to be the same. There are many parts that people have to fit together. So that idea of cooperating, it's a giant team effort, all that stuff, was where we kind of were in tech ops. But then we also met people, what they call on the floor, working actually making the material day to day, where they focus on the SOPs. What is the procedure that we have to do today? We have to do it exactly right. We have to sign off on it. We even talked to people in quality assurance, and those people were the overseers, the people looking over, did you do it right? Did you sign the paperwork? Was there something different? Making sure that everything complied with the documentation. But now I'm talking about what we learned was not really creative, even though that's what I'm teaching. I found that there are niches at this company and at other facilities where you do research and you have to be creative. And that can be everything from tech transfer, where a new product's coming in and they have to scale it up. Things don't always scale up linearly and they're not always easy to do, but you have to figure things out. So there is research there. Sometimes they're making a second generation product. So you have to look at changing those things. And then maybe something went wrong or something's different and you have to do an investigational study. So you have to figure out, can we still use this product? The big thing is that GMP production for pharma has these fixed parameters. They're established in the registration, so the government says this is the way you must do it. All right? So when you're actually doing this kind of thing, the product is the process. The process cannot change. Again, it's something different for me than what I teach. Everything is monitored, everything is recorded, and if it's not documented, it didn't happen. So document, document, document. All right? Everything is there for compliance. I learned that you learn from your job. If you get hired, you learn from your job what you need for your job, and the companies will teach you. I can't teach you everything. They will teach you what you need to know to really perform this job well. And everybody there has continuing training. Every year they have to go through and get retrained, sometimes every 6 months. So I'm going to keep teaching fundamentals, bring in some current practices, and everybody should always keep learning, like I'm doing here. I ask also people that I met, what is their advice for people in early careers? And some of the big answers were, you need to have courage to try something new. And don't be afraid to fail. can't be perfect every time. Just go and learn. How am I going to implement this in my course? Well, I'm going to put in more GMP in my lecture. I used to only just give a simple lecture on GMP. I'm going to stretch that out, have the students work on writing SOPs, maybe going through some gowning practices. And I found that in industry here and some other places that I've talked to, endotoxin tests are really important. So I'm going to have a lab on doing endotoxin tests that students might do if they go to any place in QC. I want to emphasize some more gene therapy. I learned a little bit more about it using AAV viruses, so I'm going to talk a little bit more of that. And one of the big things that I'm going to take from Pfizer is I'm going to try to reconnect with these people that I've met. So during my class time, we're going to go do a Zoom or a Teams meeting, and they're going to do a career panel for me and tell my students what it's like to work there, to do their job. Ultimately, I'd love to develop a brand new course in bioprocessing, which we don't have at the university. Have a small fermentation, half of the semester, producing protein so that they can transfer those techniques, at least know what's involved, and do a bioreactor with mammalian cells for monoclonal antibody production. And then we can also do monitoring, and we can do some QC analysis and kind of mimic what's actually done at the big pharma companies. I'd like to thank the Beta Skills program, NCBW, and especially Russ. Thanks for planning and doing all this stuff, funding through the National Science Foundation. And I really want to also shout out to Pfizer, who allowed us on site, and all the people that we met, and the people that took the time to train us, and over all these weeks took time to talk to us. And I want to say thanks to Jarrett, who was my co-conspirator here at the site, and he was with me just about every day to learn these things. And I'd like to open it for questions if anybody has any questions. Questions in here?
Okay, Jeff, you'll have to repeat the question because
Yeah.
they won't hear it online. Is there anybody online? No? Not yet, okay.
Okay, it's quiet. Well, I did have some time off, but have another minute or so. This is the fun stuff that I did while I was there. Here's Jarrett. He'll be speaking later. We went to the Bio Night. You guys were there. We went to some nice places around the area here. And on the 4th of July, I met with my family. So that was good. All right.
Okay, that's just
Go
one
ahead.
question. Were there any great surprises, things that you weren't expected or that you encountered that you didn't expect?
I think the rigidity of the way things are done and how everybody does work on a team, and it is cooperation, but they realize it actually has to be in parameters. I had a big discussion with somebody who they made too much of something. It was too concentrated. It was out of specs because it was too good. And they were kind of figuring out, what do we do with this? Can we use it? Because it was too good. That surprised me, because I'm always looking at it, trying to make something better and pure, but in their case, everything has to be in parameters. So that's a real attitude change that I have to take back with me and pass it on to my students. So thank you for the question. The question was, I'll repeat again, is what were things that if anything was a surprise for me, and it was really the attitude about having to do everything in parameters. Any other questions in the room?
Jeff, when you first came down, I remember you saying to me, oh my gosh, this is very different than what I teach. But there are elements that what you did were new to you. And are you going to be able to take those elements and put them in your current curriculum?
Okay, so for the people online, the question was, when I got here, my big comment was, this is very different than what I teach at a production facility. And the second part of the question is, how am I going to bring that and use that in my classroom? I think just trying to express the attitude that people have, everybody's working for the patient, everybody's working together, everything needs to be precise. The whole thing that, you write things contemporaneously, you put things down. You can't just take your notebook home and spend a weekend making it pretty. No, it has to be done immediately. That's a big shift for my students. What I really would love to do is all these different aspects, put them together, and again, as I said before, create a new course. That would be wonderful, I think. That would even be better than what I'm doing now. So to be able to just express that attitude. The other thing that was great is everybody was open. Everybody taught us something, even little things in just the way they did things. Pfizer has a really great culture. Every day I got an email that said, "Tell us how you're doing today." And if there's a problem, we'll talk about it, right? So they really seem to care about their employees. They're not just a juggernaut that keeps producing products. It's a lot of people there, so I definitely want to say that it was a small community, and they really did seem to care about everybody there. The guy in the cafeteria was awesome. I could never have met anybody that could remember 8 people's orders and get them right in like 5 minutes. So we were saying that he's the heart of this building. So
We've toured there in the past, and on the wall they have their values. Is that still there?
yes, it is. And the big one, other than courage, which I will emphasize to my students, the last one is joy. Do you really like what you do? I hope everybody likes what they do, and I hope all my students and all you guys can get the job that the old phrase, if you really love what you do, you never work a day in your life. So, good.
Now, if Patty remembers, because Patty was on these tours with us, she was the coordinator. But there was one value which was really interesting. I'm just wondering if it's still there. Don't be a jerk.
Oh no, I didn't see that.
Okay.
I did not see that. The things that I remember was overhearing some meetings. People would say on the meetings, remember when you're in the building, hold on the handrails, don't fall down the steps. Things like just little things that would just be like all kinds of meetings. I mean, they'd be talking about production of something else. Hold on the handrails. Don't forget to walk in the crosswalk. Just things like your mom would say, right? You can think of it in either way, that they don't want you to get hurt because that could be a lawsuit or whatever, but you should take it that they're really looking out for everybody. It is a group. And everybody was really nice. Everybody I'd see would, how are you today, that kind of thing. It's not the same in Massachusetts. So the attitude here was great. But again, manufacturing there was quite different than what I do, and I don't know, Russ, you don't know this, but I actually have some of my students who work in the Triangle area, and I met with them. They work in different companies. One works at Merck, and one works at a contract company. I was able to meet them and talk to them about their experiences, and we shared a lot of good stuff. So it's definitely an area that I'm going to recommend students move down here. It's a great place. All right. Okay. Thank you very much.
Jared has actually recorded his presentation, and so we're going to see if you guys can hear it.
Hello, my name is Jared Plant, and this is my presentation regarding my experience at the Pfizer manufacturing site here in Sanford, North Carolina. So just a little bit of background of who I am. So again, my name is Jared Plant, and I'm one of the biotechnology instructors at Central Carolina Community College, where our main campus is also found in Sanford, North Carolina. So this is actually a very local site that I've worked at for many years going to work. And it's actually my first opportunity being at the site. So it was a great experience just for that alone. Now, what I primarily teach is referred to as our BioWork course. So for those who are unfamiliar, the BioWork credential is a short-term 8 to 16-week course that was first created by the NC Biotech Center in partnership with a large group of local industry partners that basically decided what is the bare minimum skills, knowledge requirement to work on the manufacturing floor and entry level position. So we covered through the duration of the course, starting out with CGMP, good production practices, reading SOPs, all the way up to taking measurements, working with balances, measuring pH, conductivity, all the way up to working with making media, making buffers, some environmental monitoring, and aseptic techniques. So we covered all those topics in a very short term, but when they finally finish and they get that credential, it's highly regarded in our local industry because they understand that if someone has that credential, they went through all this training, and they can succeed on the manufacturing floor. So a lot of what I was focused on while I was at Pfizer was, what does it take to get someone in the door? What skills could I add to these courses to make them a little bit more prepared for working in the industry? So just a little bit about the Pfizer's site here at Sanford. So it's a rather big site. It's a 230-acre site, and in several of the buildings you have 3 manufacturing trains, primarily for vaccine production, and 2 trains for gene therapy production. So they had several products they worked with directly. Mainly the site historically was working with Prevnar, so they produced several conjugates and buffers before they get transferred off to another site over in Europe. Now in train 3, they produce Imunorix, which is their meningitis vaccine, typically sold in foreign markets like Europe and South America. Now they also have in gene therapy producing sort of to their fix for hemophilia B. And they had a second product they were preparing to produce called D&D. Unfortunately, while we were at the site, we got to experience this inevitable industry, which was it was a failure in stage 3 clinical trials. So we had, I guess the silver lining for us was going through and seeing how the employees, the environment, the atmosphere changes with, layoffs are inevitable, and how that impacts the, how the site addresses those concerns. Was very interesting to experience. And finally, other product that they mainly focused with was Lucira, which they had recently acquired. Essentially, it is a mini PCR. So it is a at-home test kit for both COVID-19 and influenza. I think both influenza A and B, which is rather impressive. So they actually produced several of the buffers, and I think some of the primers for that product, and that gets shipped off elsewhere in the U.S. So while we were there, there was hardly any portion of the site that we did not cover, but primarily we worked with tech ops and tech services. The reason behind this is that their division has a hand in almost every single part of the manufacturing process at Pfizer on the Cypher site. And because of how they address both scale up and concerns with those different areas, every single person we worked with was very knowledgeable on their area expertise. They understood the process extremely well, and were able to go into great detail on different aspects of, like, say, example, buffer prep, challenges with temperature, etc. While there, we also were able to touch on different areas such as MSAT, their Manufacturing Science Technology Group, primarily used for scale-up processes. We toured their QC labs, worked with discussing with their QA and compliance teams. They have recently gone through an FDA audit within the past month, so they're willing to go through that process. How does that look like, and what they need to do to prepare for that. We had several tours of their manufacturing floors. Again, trains 1 and 2 was for Prevnar components. Train 3 was for Imunorix. And then finally got a tour of their gene therapy suites for train 1 and 2, which train 1 primarily was for fitness. And along with that, we were able to actually go through their new employee orientation during our second week there. And that was something I was actually looking forward to, primarily because I actually had to work at this site directly, work at Pfizer, and I can now tell them what it's like to go to an employee orientation and their accounting qualification, what that experience is like, because again, most people, the main question I get from those taking my courses are, what is it like in these facilities? Right? So that's been my biggest takeaway while being there, is the atmosphere itself. So I already touched on it a little bit just then, but what am I really getting with is understanding what it's like to work at not just Pfizer, but working at these local industry partners, and what the environment, the different areas that someone can work in, the culture itself, and what requirements do they need, and what they look for in these areas. So for example, some areas that they do is like manufacturing, eventually they may want to go into tech ops. If they have some more lab experience, they may do better in QC and maybe eventually do QA. But, and that's a dynamic I didn't understand before, prior to my experience. And along with that is understanding the dynamics of these departments. So tech ops is, again, a very good department for us to go through, as, again, they had a hand in almost every single aspect of the site, and seeing how these different groups work together, what area specializes in maybe one technique versus another, the difference of the MSAT labs versus QC labs. But along with that, we had a couple points that someone pointed out that I hadn't really taken into consideration before that I want to incorporate in my courses. Of course, there's things like attention to detail, our documentation, which I already covered, but a couple things. So this site specifically, since they work a lot with tetanus and live viruses, they have a large portion of their materials are single use. We discuss it in the viral course, but we don't go into anything such as like working with those connectors and following teammates. So I feel that's things we can easily incorporate in these courses to give them a chance to actually work with the material before getting to the floor. And also maybe discuss graphing and trend data. That was something that was brought up to us towards the end, that someone said that understanding like ELISA, PCR, those informational assays, not being able to actually perform them, but understanding what those mean and how those data trends influence decision making. So they said, why are we doing this process? Why are we making this change? Why that matters. That definitely is something that I feel like we can easily incorporate, especially in the earlier portion of those courses. So I just want to say that it has been an absolute pleasure working with Pfizer Sanford site. I'm very thankful I was able to join this opportunity. Thank you, Foresight Tech, Beta Fellows, for funding this, coordinating this opportunity. I do also want to say that thank you to Jeff King, who was with me at the Pfizer site. He's very extremely knowledgeable on these lab techniques and was able to ask questions that I wouldn't have been able to even come up with. That definitely was able to give us a little more insight into various processes in different parts of the manufacturing floor and labs. So, great thanks to Jeff. And then finally, I'm extremely thankful to be able to take back this experience, have these stories, scenarios that I can bring back to my students and share with them. So again, the end goal for us is to get them a job working with our industry partners. So that's it for me. Feel free to send any questions to my email, jcla598@cccc.edu. I'll put it in the PowerPoint as well. And just want to say thank you again for this opportunity.
Ryan, you love this stuff?
Yes.
That's great. Jeff, anything, because Jarrett isn't online?
Should I stand up?
Yeah,
So,
come to the mic. You guys work hand in glove?
we were hand in hand, and it was great working with him because he was teaching at a different point than I'm teaching. I'm teaching something, like I said, more research. He's teaching people directly to go on the floor. So he saw students that actually were his students there, which was fantastic. And the idea that he said he was able to go through the training that he's teaching the people about directly. So he was very connected with everything that was going on at Pfizer. So to see that aspect and to realize that all these different layers have to go through. People from, everybody working in the office to everybody working on the floor to people dealing with vendors to see if they have 10,000 grams of whatever, and then having that verified and going through. It's a whole big team effort. So it was good to have his perspective and what his students are doing, and to really try to give that perspective to my students. So I think it was a good give and take.
Excellent. I remember the question I wanted to ask. We have toured Pfizer. We haven't been there for a couple of years, but there was, when we go into some of these big production plants, some people are doing paperless documentation. And what's the status at Pfizer right now?
It's a little bit of both. A lot of their material is on computer. They're monitoring all of their processes in the production area, all on the computer. They can literally click a button and a valve opens and things get pumped from A to B. But at the same time, there are some documents like the SOPs that people on the floor have there as a reference that they're reading through and either have to sign off on or go on the computer. Everything is scanned and everything is uploaded, so all of their documentation is on the computer. But some things are still being used for paper, particularly, and you have to have something in front of you, SOPs. I mean, when you're going through manufacturing, people are carrying their computers around doing things on there, so a lot of stuff is directly on the computer as well.
Great. Any other questions? Good.
I mean, good experience.
Thank you, Jeff. Thank you, Jared. Olivia, you're up. Olivia is a student at George Washington University. And she came to us early in the summer. And she had approached us, I think it was back in February maybe. And she said, I'm looking for an internship. And we said, well, hey, well, let's think about it. And so we said, sure, we can use you. So but having had kids that had done internships, I wanted to make sure that she had a real internship as opposed to doing something that really wasn't going to enhance her education. And she was a really good fit because she's doing biomedical studies. She'll talk to you a little bit more about that. But she was a great fit. And of course, we're doing what we call biomedical emerging technology applications. So it's a really nice fit there. And then, so we had something called the Biosciences Administrators Program, and she'll talk to you about her experience doing that. But Dr. Green is here, and Dr. Green is the former president of Foresight Tech. And he had moved over to the Institute for Regenerative Medicine approximately 5 years ago. And most recently, he was able to, with the team at WFIRM and some of us, acquire an NSF engine grant. So it's a rather large grant. It's the first 2 years are about $15 million, but the potential for the grant is $150 million, Dr. Green. And so, through the team, through his efforts, and Dr. Tella, they won the grant. And so we had to handle the workforce portion of the grant. We had to plan it, we had to get it going. So Olivia's going to talk to you about her BAP experience, and she's going to talk to you about her engine experience. Would that be a good summary? Okay, thanks. Olivia's been great.
All right, so yes, I'm Olivia Brown. I am a junior at George Washington University studying biomedical engineering, and I have been doing this internship working on an engine and with respect to workforce. Okay, so naturally I learned a lot about regenerative medicine, and that it's a very multidisciplinary field, and the products that result from RM include cellular therapies and agents that trigger natural pathways of regeneration, and complex tissue engineered products comprised of organized living human cells And the goal is to replace and restore function to tissues and organs compromised by injury or disease. These are some pictures I took at WFIRM during our tour, and that I thought they were pretty cool, so I did them. So to start everything off this summer, I looked at the Piedmont Triad Regenerative Medicine Engine grant document and found particularly workforce and education goals and plans. And at the center of that is the ITEX, the Innovation, Translation, and Education course, that are facilities established to focus on different unmet needs of RM. And the collaboration between these ITEX and the industry partners is an important network to address the many goals of the engine that I've been learning about. And the overall engine goal is to grow the RM industry in the region. So, some of the information that I've gathered to help us with our goals in workforce development have been the education partner programs. I found STEM and life science centered degrees from the 4-year and community colleges with skills learned in this path and the paths. And also some of the ITEC summer programs, which include high school summer programs, training, fellowships, and internships. I found some economic development partner programs that were mostly entrepreneurial / business mentoring programs, with some of them including STEM relevance. The partner company sizes for the PTRME, I collected employee numbers and how many specifically in the region. I also found I compiled the number of students in life science and related degree paths for Winston-Salem State University, NCANT, UNC Greensboro, and the regional community colleges, and was able to have a rough number of the number of students in the region in STEM. And then some job skill sets. I looked at some life science jobs in the region at different levels and compiled the different skills needed for some like entry level and higher level jobs. And all of this info that I gathered was important for some SWOT analysis where we highlighted 3 of strengths, weaknesses, opportunities, and threats for workforce development in the region, and some information was used as supporting evidence in the analysis claims, as well as risk analysis and mitigation strategies, which was the most recent project. I'll cut off a little bit. So the Beta Back program was early on this summer, and we toured different sites to learn about skills and occupations in regenerative medicine and biotechnology. So first we toured WFIRM, and during this visit we were able to see the different projects going on and lots of equipment used for these projects. Everything was very cool and innovative. And I learned that they have lots of people from many different academic backgrounds, including undergrads during the summer and PhD students with regenerative medicine concentrations. And the tours were able to give us insight on new skills and occupation paths in biotechnology. And it was great to learn about the current happenings that are in the workforce. These are some pictures that I took during the program. And we also toured BTech at NC State, where I compiled some of the skills that are learned during the courses, and we got to see the facilities, all the equipment used, learned about lots of the training, and I added skills to a larger list for the PTRME. And that's it. Thank you. Any questions?
I have a question. Kind of goes back to the beginning when you were touring the regenerative medicine. You showed some pictures that looked like ears.
Yeah.
Can you just address what level they're at now? What's the cutting edge? Are they taking cells from humans and growing back parts like ears, or is
These,
it all in mice?
yeah, sure. The question was about where they're at with growing these. These particularly were for cosmetic purposes, and so they're working a lot with just people who have maybe lost different parts, and so they 3D print things to gain confidence.
So they were actually 3D printing some of this rather than just growing in a shell or a mold?
Yeah, I believe this was just, they had a bunch of the different things that it was made out of, but yeah, this was just for cosmetic.
Jeffrey, the way that I understand that is that those are scaffolds that they're going to 3D bioprint onto and then grow the cells from there.
But it's actually 3D printed and then they grow out rather than the scaffold on the back of a mouse.
Right, the scaffold will hopefully dissolve once it's grown to what you want it to be.
Yeah, like stitches. Absolutely.
That's the way that I understand it, and I believe they put some on, haven't they?
Yeah, the Institute currently has about 16 different tissues and organs that are actually in patients now.
Excellent.
Now, they're not commercial in the sense of being on the market for placement, but they're in clinical trials at various levels. Some of them have been in patients now for years to track them. So the cranial facial work includes like the ears, nose. A lot of that has been sponsored and supported by the Department of Defense because of battlefield injuries that oftentimes require, or there's a need for that kind of reconstructed. So, yeah, they're using bioprinting, they're developing scaffolds, using nanoscience and nanomaterials for scaffolding and for creating what you see there is the models for the ears.
I know they're at the level of skin and cartilage and things like that.
Using much as increasingly personalizing using autologous cells for actually growing the replacement parts.
Yeah. Thank you. Both.
Olivia, what was your favorite part of your experience to date?
I really enjoyed all the tours that we went on. WFIRM was very interesting, but I kind of felt connected to our Cook Medical tour just because I was listening to all of the different people who were bringing us through the facility, and I could apply different things that I've heard in the classroom and through my education, and was able to see things that were actually happening in the industry, not just be on a slideshow. So I really enjoyed it.
So if I heard it correctly, Cook Medical?
Yes.
And your current degree that you're seeking is
Is biomedical engineering.
biomedical engineering, so yeah, that makes sense.
Very cool. How about a big round of applause?
Great, so I'd like to introduce Aaron as he me introduce Dr. K. He spent the summer with us. I worked for Novex Innovations here locally in Winston-Salem. Aaron was a great benefit to our company. We hopefully had a mutually beneficial arrangement. He was influential in helping us get UPLC at our company up and running this summer, and we greatly appreciated his time. He got to review many of our SOPs, go over some training, and I'm sure he'll go into some more detail on that. Thank you.
Thank you, Bob.
Hi, thank you. But are you able to see my slide on that, or you only see my background that's not over there?
Desktop only.
Oh, that's not good. Let me share the client screen. Is that correct now?
Yes.
Yeah,
Yes, okay.
you're good to go.
Alright, cool, thank you. Hi everyone, I'm Aaron Kamajaya. I'm a faculty at Los Angeles Pierce College, mostly teaching biotechnology, and today here's just a little presentation on my experience as a beta industry fellow at Novex Innovation. So first of all, I want to show you the outline of my short presentation. What background first, who am I, why am I doing this, and then we'll go into the what do I learn, what's that impacted our biotech program, revision and making improvement, and then last but not least, closing remark. So let's get into that. Who am I? Me, that's my background is purely research, coming from academic research training. Mostly, if you look at on the right side, it is sort of like a summary of all of my research background, working with bacteria, Caulifacter Crescentus, doing some immunogenesis, looking at cell cycle, and then I did some structural biology as a PhD student, working on mechanosensitive channel or piezo channel, using things like using the technique like X-ray crystallography, single particle, IBM, having experience expressing protein in different expression systems from bacteria, yeast like Pichia, insect cell, as well as mammalian cell. And then after that, once I finished my PhD in 2017, I worked as a part-time faculty while holding a part-time appointment, research appointment at Caltech as well, working on biosensor with protein engineering to develop biosensor for drugs, particularly in the company. So, and then from there, shortly after that, 2018, I landed my tenure track faculty position, in which we actually hired me to teach microbiology for pre-health students like nursing, dental hygiene, things like that. But in addition to teaching microbiology, somehow I don't know why I decided to create our biotech program, biotechnology training program, which is here. Basically, this is a snapshot of our biotech program. What make our program unique is that we actually offer authentic research experience for students, where every student work on an authentic research project, and then they get to cross collaborate with their classmates. The project involves the protein purification. You can see at the bottom left corner in our student purified protein. Over there using our FPLC machine. And then we also did some work on nanobody engineering. We have our little lyophilization machine. This is not an industry size collaborative machine, but nevertheless you can see at the center bottom over there, there's a lyo machine that we have. We have this automated cell protein expression using the cell growth modifier one on the top left video over there, and several other PCR, things like that. And then at the end, our student presented the research, the finding in angle bullshit, which is one at the bottom right corner there. So that's basically a summary, a snapshot of the our pilot program. And the question is, why am I participating, or why did I apply to the Beta Industry Development Program? To get this internship. Well, I think earlier this year actually, spring 2024. Well, actually, let me back up a little bit. Our program actually grew very rapidly. We actually grew from one offering, actually one cohort entry per year into that was back in 2021. We only have one entry for the cohort for that year, and now in 2024 we have at least 4 entry in this 2024-2025 cohort. So there will be 4 entry in the year, including 2 enrollment. So we really grow rapidly, and our class is always full. So we always started our cohort with full enrollment with some waiting list. So just to give you perspective, like in our size is 20 students, so we always be able to fill our class. For the fall 2024, not only we fill the class, but we also have 14 people in the waiting list. So that's almost like another separate cohort right here. So because of that, we gain a lot of attention from our upper management. Not only we deliver the enrollment, but also we bring in money. We get the NSF grant, we get other local regional grant, and that's sort of like what makes our program chosen, I think, by our upper administrative people to go for the baccalaureate degree. So when in California, there has been a move to allow the community college to offer baccalaureate degree program, and our biotechnology program was chosen by our administration to be the very first one for our college. So we did apply, and then just earlier this year, our program get full approval from the state, and we need to offer it. It's a good news. I get a lot of congratulations from my colleague, my dean, VP, but for me, I'm kind of nervous because how am I supposed to teach this? My background is fully research, just plain research academic. I never actually worked in manufacturing facilities. So this is where I started looking for opportunity, and I saw Russ post in LinkedIn that it's just want to do internship for 8 weeks in a company. So I signed up. I signed me up. So that's basically why I'm doing this. And then so I'm actually very grateful that Knowledge Innovation really to take me in and show me the best practice in manufacturing, biomanufacturing, and even just the standard clean room, the operation, and such. What do I learn when I'm in Knowledge Innovation? Here's the slide. This image is not taken from the Knowledge Innovation, just to let everyone know, so to make everybody nervous that hey, I'm taking picture from the company. This is actually taken from the feedback capsule facility. This is their fill and finish machine. This is actually a very common machine in the manufacturing facility. They have Knowledge Innovation, but this is not the exact picture. The exact equipment and specs. So what did I learn? What did I see and learn during my time at Knowledge Innovation? This is just a short list of what I learned. I feel like we learned a lot more than this. It's just something that sort of made me sort of wow, like aware. So number one, that the GMP facility is highly regulated. You can't just wander around and play good in the room. You can't just do that, right? Everything is very regulated. every single procedure is formally written in an SOP, which is, this is kind of very different than academia, right? For academic, like myself, if you want to make media, let's say LV broth or something like that, you just Google the recipe or look at that from someone's recipe, and then you start making it. Just start making it and then mix it. But here there's a SOP that you need to follow exactly how you're supposed to mix them, what else you need to do, and there's forms that you need to fill, like all documentation. Also, like number 3, all documentation have uniform format as a very unique control number for each document, so it's maybe easier for you to index it and find it in the future and revise it. There's a lot of validation and there's huge, very huge traceability. So for everything that is done, you have to know who did it and when was it done and so on and so forth. So you know exactly, you can trace it back. How is this process done? And then I also get a chance to look at the gowning process, learn about it, and then the process qualification process, as well as improved behavior, which is I think something very unique experience. But it is the reason why I feel that I can always learn by reading something from the textbook, but nothing compared. Like you actually be there and see it live, how people actually do it. It's even how they behave in group. So it's very, very eye-opening experience for me. Also, I learned from the clean room design as well as the basic cleaning routine. Like you have to mop the floor, for example, and cleaning the bench. Periodically with something with a set of different what is it called, disinfectant every week or something like that. That practice is something that really opened my eye when I see that. So from this alone, when I was there, it sort of inspired me to restructure our biotechnology program. So just a few things that I want to share, things that I started to create in the process of restructuring our biotechnology program is number 1, we create a formal SOP for every single process that we have. So this, so in the morning, I was in the NOVEX learning about stuff. After coming back home to the hotel, you can ask our other fellow Fatima, Rafi, that whenever I'm in the hotel, I could go to the gym, eat dinner, or work. So this is a part of this work that I've been doing, restructuring all the SOP, rewriting all the SOP, putting the control number, things like this, even for simple stuff like preparation of buffer, So, and then not only that, I also designed some, new course which again has to be approved by the college universities about the clean room and manufacturing, setup manufacturing, something like that. And I also designed the environmental monitoring activity for our introductory biotech program. We plan to acquire this sort of a device for the viable and non-viable kind of particle counter. But yeah, again, I'm gonna start. We plan to purchase this soon. And then last but not least, I also sort of like in the syllabus and everything, we start to introduce student the terminology, industry terminology. It's kind of like instead of calling like practical exams, instead of calling them like practical, we call it qualification. So then again, we start to implement this, introduce this terminology even in the syllabus and the entire class. So instead of calling it exam, we call it performance review. So something like that, just a simple thing, but I think it will make the student aware of what is this qualification all about, right? So when I was there also, I had a chance to talk to Doug, and I asked him about the design of the clean room. We want to build our clean room, so what's the best way to do so? So he shared a few tips and sort of like how they design their clean room over here with covered wall and everything. But he also recommended me, hey, perhaps you can visit the Capstone Center at NC State. That way you may get a chance to see how an academic institution training program will design and build their clean room. So that's what prompted me to ask Russ, hey, can we get a tour to the tech facilities? That's one that you can ask, and surely we find Russ able to get us a tour to the Tech Campus Center. And I actually learned about quite a bit more how they run the program, what sort of like technique they teach, and so on and so forth. And that's also gives us a glimpse of various clean rooms that sort of inspire me to design our own clean room. And this is kind of thing, I'm really stubborn individual, pretty much like stubborn, but people, like a lot of these things I've heard my colleague, like Jim DeGlow, for example, I got a chance to tour his facility. I also talked to, let's say, Matthew Bryan, where we actually look at, I've seen actually the SOP like this is standard SOP like this, formally written. I've seen it once, but then it's, at the back of my mind is that really what the industry is doing, right? So this is what when I actually did the internship here, it's actually like, wow, this is really what my colleague is doing is actually what has been implemented in the industry. It's like the best practice in the industry. So that's, again, this very, very helpful function for me. So from my tour to NC State, it will help me design our new clean room as well as the Mammalia Counter room. So yeah, and then in addition to that, at Novak they actually did me this independent project, I call it project of EPLC, where they have this emails machine that's been sitting there, and hey, do you know how to make it work? Okay, I've never used that. If you trust me that much, I mean, I work as we have at PLC for more of a purification, but I never actually worked with analytical part, analytical machine like this PLC. But we tried. We did some work on it. Again, this is not the actual image from Novak's innovation. This is the thing. We actually are kind of debating whether I should buy this one. Our training program, but in a way they have let me have fun trying out this machine and sort of like looking at the different standard, what is the upper limit in terms of the linear range of the machine, doing the asymptotic injection and so on and so forth. So it was quite a really interesting experience. I know Ryan earlier mentioned about I'm helping them with this machine and all that, but I also learned a lot just by playing around with the machine. Again, I never used this machine, and it's also really valuable learning opportunity for myself, just to get to know the machine, how it works, how to set up the protocol, how to integrate the chromatogram and things like that. So it was quite interesting. And I think that's all that I have. I'm sure I'm more than this right now. Yes. So here's, I want to thank everyone, the Forcing Tech staff, particularly us, that make this opportunity possible. Mary and Patricia for helping us, all the fellow, and also my Winston Salem, before dinner party, and event, the four of us share the car, we went quite a few places together, and people at Northwest Innovation wanted to play Brian that showed me around the practice in the manufacturing facility and everything else. Doug and Jerry for allowing me to stay and good discussion on clean room facility and Elena and Emily, the manufacturing technician that sort of like, babysit me pretty much. And then and the BTech custom facility center, Christy and Michelle. And that will be all. And thank you very much.
Questions? Brian, do you have any questions for your student?
No, I just want to say again, we really appreciated having Aaron on site. It was a huge benefit for us having someone working in the UPLC, like you mentioned. And I hope it sounds like it was beneficial for him as well, just to see our processes and see our clean room design. Working together was a great experience. I really appreciate it.
So Aaron, in case you couldn't hear Ryan, he was thanking you and he said it was a great experience.
Yeah, thank you.
Could you hear him?
I can hear you, but I can't hear the audience
Yeah.
talking.
Okay, great. Any other questions? Jeff?
No, we talked at Bio Night, and it was very nice to meet him, and here's the programs.
Not a question, just a reminder, I want to remind him to share whatever he creates with us.
Okay, good. Okay, Aaron, thanks so much. We really appreciate it. I'll see you next week in Kansas.
Thank you. See you.
Okay.
Thank you, Kansas.
Bye. Bye-bye.
Bye.
Hey everyone, my name's Zach. I'm a fourth year PhD student at WFern. So what our project is working on is developing models for intestinal cancer that are going to be able to be 3D printed at a high throughput rate to try and study different disease models and to create models for personalized medicine. And so what we did with Rafi most of this time is we kind of were, well, she'll say we're all over the place this summer, but she got to learn a lot in a lot of different areas. So we made bioinks, we chopped intestinal tissue, which she loves, and we did a lot of decellularization and preparation of the material. The major goal of the project is to incorporate decellularized intestinal tissue from either pigs or humans, and kind of incorporate those proteins and signaling molecules into our organoid system so that they'll be more physiologically accurate. Rafi has honestly been a blessing. I'm kind of annoyed that she's leaving, so and she's been absolutely great. I was not expecting anyone so awesome. She's just been an incredible help this whole time, and I'm hoping she learned as much as she was helpful. So.
And hopefully this is not over. Who knows?
Yeah.
Thank you, sir. So you're going to see different presentation than Aaron. He's like much more structured. I'm not. I'm more freestyle. I got introduced to these programs 2020, right? I gently was like a volunteer to become the biotech coordinator for FBCC, which is Bronx Community College where I teach. And I don't know anything about biotechnology. So I was like, oh, great. And I was like, oh, I'm gonna learn so much stuff and I'm gonna change so many things. And I didn't know that you don't change anything when you're working in academia because there are articulation and a lot of other things. And then obviously, COVID, hello. And that's actually when I discovered InnovaBio, and I started with the program that Rasa had created for the leadership. We did everything online, but I started learning a little more about biotechnology, and then I'm just addicted. I'm back like in North Carolina pretty much every summer. And I saw this program last year and I was super interested, but I was already busy doing other things. So I had to do it this year. And the reason why I wanted to do it, well, I'm just a nerd, right? We're all scientists here, and we're just interested in anything that has to do with science. We kind of want to figure out, hey, can I learn something more? And whatever it is going to be interesting. It doesn't matter what. I also really don't know anything about translational research or industry. I learned a little during these programs over here, but you know, I'm still kind of a newbie because I've never really worked in the field. Obviously, because I am an educator and I'm the coordinator for the program, I'm always looking for different opportunities for my students in terms of job, yeah, the workforce part, but also internships and so on. Working in a community college, you really want to be able to build up your students in a way that they feel comfortable and confident applying for internship and things that they would normally think they cannot actually do. So if you know, you can share with them and you can build this kind of confidence. Also research opportunities. Low-key, I wasn't really thinking about that, and then I realized when I was working with Zach and with his mentor, that that's actually a possibility. So I'm like, okay, cool, that's even better. I don't know exactly what I can offer, but I'll take it. And obviously the main thing is really to try to think about like practical activities that I can incorporate in the classes without driving everybody crazy, like you can't do this, you cannot make changes, but just like specific activities so I can actually allow my students to learn some of the things that I've learned on the field. And then talking to Aaron, who's a different level than me, is like, what about grants? And I'm like, what? So I don't know, I'm thinking about it. All right, so that's the one. Well, I actually ended up at WFUR, which again, I was like really interested because, come on, Institute of Regenerative Arts. I wanted to be there, and I wasn't sure exactly what I was going to find, right? Because I still, when we visited, it still made me feel a lot like basic research, some of the things that I was interested, so I wasn't exactly sure what I was going to learn. And I'm not gonna tell you guys, like half of you actually are from WFUR, and Zach has already introduced some of the stuff, but obviously tissue engineering, regenerative medicine, and I kind of lifted this from the page because I wanted to remember all the different things. But again, it's really more translational. So it's been in the lab, but you're not just thinking about how does this work, but how can I actually produce something that can be used by clinicians and by people and possibly can be scaled up. I ended up in the lab of Dr. Shay Soker, working on organoids. Zach already just gave a little tiny bit of the explanation. This was my team. Obviously, there's Zach, there's Sach, and with Pratima, we were like, partner in crime throughout the summer. So I consider I'm part of the team. And again, I was working on 3D tissue construct organoids, which I kind of heard before, but I didn't really know that much about. And again, I was just thinking about regenerative medicine. I wasn't really thinking that much about the other part of the clinical stuff, so I was very happy that I had the chance to learn this stuff. And as Zach was talking about, it's like, we are moving toward personalized medicine. We want to know how to treat patients in an effective way. So what's the best thing than actually be able to look at the cells of the patient that has the disease, and instead of testing the drug on the patient experimentally or on animals, which we know don't give a really results that are quite often like true to what the drugs can do in the humans, why can't we create an environment, a 3D environment, that where the cells can grow, even different cell type that can be found in different tissue, and then we can try the drugs on there. So I'm like, wow, that's actually kind of interesting. And there's a full institute that now has been created for that, so that's even more interesting. So what did I actually do that made me super happy? Yes, I chopped up a bunch of like intestine from pigs. It was interesting, we had to optimize that one too, so it was actually an interesting thing to do. And I learned how to extract ECM to make organoids. So I never really thought about it, right? You have these little gelatin things, but I'm like, wow, it does make sense. You can create this 3D structure, but it makes much more sense if you're actually extracting the protein from the specific tissue that eventually you want to treat. So in this case, we were looking at colorectal cancer. So we were actually looking at the intestine of the pig. I didn't really make the organoids. He made them with or without cells, and we did some 3D printing, and then the 3D printer was like, I'm not feeling it, it's like the summer, went all European, and decided to just take the summer off, but we still had the chance to try a lot of different things. And to me, it was actually kind of interesting to see what went into what I would call optimizing a protocol if I was doing basic research, but it wasn't really optimizing, right? It's actually really looking at how can I use other people's information to make my protocol the best for the specific things that I'm doing, but also thinking in terms, I want to scale up. This is gonna be a product. It's not just going to be like, oh, I'm gonna make it work for my experiments so that I can get a figure, do my paper, and then move on with my life, which is, I think, what basic researchers do. Maybe I'm judging them. So I did work like what you're seeing here is actually how the ECM looks after it's been extracted, dried up, decellularized, defatted, which is like something that, again, once you know it, you're like, well, obviously you want to remove the cells and you want to remove like the lipids, but you know, I never really thought about it until I was doing it. And this is actually something that we were doing even if we didn't have the 3D printer. So these are the organoids. They're very beautiful because they're actually keeping their shape. I did have pictures of them with like in the media, but they were not as pretty, so I didn't put them. Something else that I actually did, because somehow I did some of the training, which made me think about other things that I can do with my students in terms of like, hey, what if they want a job in one of the core facilities, which again, WFIRM has a lot of those, was some histochemistry. I did some staining of cells, but I never really did the full like histochemistry in my experience before. All right, so we did a bunch of different things, as Zach was saying. What am I actually bringing back, and how I'm going to implement it? Well, first of all, again, incorporate activities in classes that have to do with protocol development. We are updating our classes at BCC, and we are trying to push a little more the critical thinking, students actually thinking about setting up their own protocols and things like that, literature readings. But I never really thought about looking at the materials and methods the way that we do when you're doing like research, but also in terms of like, okay, obviously there are a bunch of different ways that you can decellularize tissue, for example, or remove the lipids, right? You're not going to reinvent the wheel, but at some point you're going to have to choose which one works better for you, because if we're thinking about the product, there are so many ways that you can extract DNA, but we have a lot of companies with their own specific recipes that they're not going to tell you for any one of their like little like, you know, mystery bottles. So I want to communicate this with the students because a lot of them, they have this feeling like they don't know if they want to do basic research, they want to do something that is a little more clinical. Some of them are interested in business. And I'm like, well, if that's the way that you're thinking, and you're thinking, oh, I'm gonna get like a PhD, but I want to have a startup, I want to make my own products, you need to start thinking in this way. You need to start thinking about like, well, how am I gonna go about doing that? So I'm trying to think about like different activities, starting even with the literature, where they're actually looking at the materials and methods, and they're looking at some of these papers where they're trying to optimize and refine some of these things. I'm going to try to figure out how to add some histochemistry activities. They're not super expensive, so I think I'm okay. And we do have a lot of things that can actually fit, and I can make it sound like I'm actually covering other stuff that is in the classroom. In terms of research, again, I was a little surprised. I don't like WFIRM, BCC, I don't think there's anything that can actually go together. But I found awesome people. I don't know what I can offer, but I'm definitely going to think about it. Because I would like to keep this conversation also because it's interesting for the students to do some research that it's meaningful, right? Someone is actually going to use it. They like to learn, but they also like to know that whatever they've done is contributing to something. And then, thanks to Aaron and those long conversations where he was like stopping his work and actually coming back to us and having tea, you know, we had to book his time. but then we could talk about a lot of interesting things. He was actually talking about grant ideas, which I was truthfully not really thinking about, and no, I'm not gonna go this year because the deadline is in October, and my brain cannot think that fast. But we were actually thinking about that in terms of like what he did with his grant, creating activities that are specific to tissue engineering that then can be shared, and there can be a grant. And again, now that I've done some of the tissue engineering, I actually do think that I can incorporate activities or I can come up with projects that the students can do, and potentially when we have something solid and cheap, because it's community college, we can actually be able to share with the rest of the world. Yay! And that's pretty much it. I don't have everybody to thank you. I just put Millie, did I say the name right? So this is actually Zach's dog, and this is one of my turtles. And I just put it because I think everybody's super stressed out, and we should not forget that we have people that support us, together with Russ and the MSF and everybody else. I'm very thankful to everybody, but we should also remember our support like that maybe we have at home and keep us safe when we're stressing out with work and all the other things that are going on. Ta-da!
Questions?
Perplexities.
Questions? Must be a question.
Yes, I know the question is like, that's the name of the turtle is Timmy. He's a male, right here, slide there.
You have two?
How's
Fine.
Timmy doing?
Tim is doing fine. He was actually like finished to eat his food this morning, and then he was upset that I was leaving.
Ask about Dora. Is she still bad?
She's still
She
mad.
has a
They know everything about my turtles, I'm telling you.
She has a turtle who gets mad at her when she leaves. So.
Yeah. She actually gets really upset about it.
Did your apartment smell like a swamp when you got home?
It's a little like that now because I didn't have the chance to clean the filters. But you know, usually it's actually pretty good. I figured out, I'm a scientist, I figured out the way to actually live with all the turtles in my house in a New York apartment. No, nothing.
There must be a question.
Make us happy. Hi.
Some technical questions. So
Okay.
I've done some organoid stuff for breast cancer, and they're very difficult to keep those cells alive. Well, they make some little packages. In your work, you're mainly working with removing cells and then reprinting them, or were you using that as a scaffold where you remove?
So we were actually getting the ECM, the
Okay.
proteins, and then we were mixing it with some other proteins that can polymerize. So Zach project, without going into detail, because that's his detail, is to actually be able to extract the protein from the ECM and then treat them in the way that they polymerize as well as possible so that the organoids actually keep their shape. But what we were actually doing, we were mixing in HCT116, which are colon cancer cells, and some of the fibroblasts. So we were doing different things, but we were actually putting the cells in the organoids to see if the organoid kept the shape, but also if the cells survived. And that's actually one of the pictures with the cells surviving, that's the green stuff, and these are the cells dying, that's actually the red stuff. So
Live data assay.
yeah, and so the idea is that if these can actually be scaled up and it's like optimized and so on, you could take the cells from the patients and grow them and then put them in the organoid and print them out and then treat the organoid with whatever drug and so on that you want. So
for the company, is that going to be, you're looking at the matrix being the product or the cells or the combination?
the matrix.
The matrix. So have you tried any of the commercial, have they tried any of the commercial available, like Matrigels and things like that?
I mean, I'm sure that that's something, but so the idea is like we do have product, right? But they are not specific products to the specific organ that you're treating. So if we are talking, I mean, ACT116, they're cancer cells, they've been in like forever in culture. We know that some of these cells, they're gonna survive even when you want to kill them, like
Right.
they're gonna survive.
Like human cells.
Right. But if you are actually taking something from a patient, you really want to have the best environment and the environment that is truer to where the cells are coming from. So in this case, and again, it may be correct or not, but if you're looking at liver cancer, you're going to try to get the ACM from the liver. If you're looking at like a colon cancer, you're going to try to get it from the large intestine. So the idea is that the mix of protein is going to be different depending from the tissue where the cancer is. So you want to create the best environment for whatever cells you're testing.
One follow-up question. You were looking at pig tissue. Was that any particular like strain, or was it genetically modified pig or anything?
Straight up from the slaughterhouse, that's something that I discovered that when I was reading the paper, and also when we got this like, oh, they killed the pigs today. We're gonna get some like allergic testing.
Yeah.
I don't know. As far as I know, we are just like also from the other paper, everybody's getting the tissue from the slaughterhouse. I actually do believe, however, that the first ECM that we were using was from humans,
Yeah,
not from the slaughterhouse.
Yeah, we use human and pig, and it's not modified at all. It's kind of regular tissue.
I'm just asking, is that a potential for more success if it was more humanized?
Yeah, so we currently are using human small intestinal tissue right now. So this was all to try and like kind of create our initial model, and then we'll be moving forward with kind of seeing what we have available.
Sounds like great work, and I wish you luck.
And again, like Zach is doing some modification to ensure a better polymerization because we want to have these organoids actually surviving. I am also going to start like a t-shirt business with images from science. You may or may not notice, but this looks like a shark, and that's a shark after it's eaten. So this was for Shark Week. So if you have some interesting like images that you're getting from your research, feel free to send them to me, and that's going to be on the side.
I've seen companies that put out calendars like this. So
I think I'm going to make more money with the t-shirts.
I've seen somebody who makes clothing with images.
Yeah, that's what I'm thinking. And I am like, I discovered, we were at UNIH last week, that seemingly that's something that moms at home have. They bought like the complete out t-shirt. So that's the other thing that I came out from this summer.
The side hustle.
The side hustle in case like, this gig with the biotech doesn't work out.
All right, thank you.
Zach, thank you for
Thank you
W
very
Firm,
much.
and
Yeah.
Dr. Green, thanks for W Firm, and Rafi, thanks for a great presentation. By the way, she flew in specifically
Yeah.
from New York to do the presentation.
Wonderful.
So now we have Yvette Mumford online.
Okay, so I'm really hey guys, so if you want to say something to me, you gotta shout. So, like Russ said, I'm from Texas. Work at the Texas Academy of Biomedical Sciences. We're an early college high school, and that's where I teach biotech. I also teach medical interventions, pathophysiology, medical micro, and AP environmental science. And this summer, I was able to work with Javara, which I had on my original slides. I'm not quite sure why it's here, but I wanted to thank them. I worked with Dr. Colleen Cannon most of the summer and her team. And of course, wanted to thank Russ for setting everything up. Okay, so Javara is it's a really young company. It was established in 2018, and it provides clinical research access for patients at the point of care by embedding staff and infrastructure into large healthcare organizations, and that it is resulting in greater access to cutting-edge innovations while expediting FDA approval through reliable, robust data. It's kind of important to note that they gather data and they facilitate the process between the pharmaceutical company and the FDA, but they don't analyze the data themselves. So they kind of gather it, and they're kind of the middle person, but it allows things to go faster. So, Javaris is nationwide, but they have like regional leaders who head up teams that work to match clinical trials to the medical clinics. They have certain clinics that they work with. They need to make sure that the clinics have enough patients that meet the criteria. And so with each test, there's about 30 inclusion things that they want a patient to have and about 30 exclusion. So like they might say that they want somebody to have high blood pressure, but their BMI can only be within a certain range. And so they could have qualified for one and then disqualified for something else. Javara has direct access to patient records that are within these clinics, but they're working on an in-house app, and the in-house app is called Genesis, and it's going to provide faster, easier access, not just to the records of the patients, but say that a patient got disqualified from one study because their BMI was too large or too small, either way. And then the next time they want to test the drug again, but this time they want to test it on a different patient group, they can be like, well, now we want people that have this other BMI. So then they can just grab those really easy out of the app that they're creating instead of having to go through all of the patient records all over again, which is very time consuming. So this is definitely like a production amplifier. And it's still work in progress, so meantime they have about 3 different external applications that they use to gather all of that information from the clinics. And sometimes those other apps don't give you all the information you need, or they just give you way too much. And so you have to go like kind of search through the weeds to find what you really need. And they're just trying to streamline it with their own in-house app. So they gather data from the trials, but like I said, they don't analyze the data. They send all the raw data back to the actual company for them to analyze themselves. But it just allows them to do it faster. So one of my favorite departments, I work with Miss Ashley, but she's located there in Winston-Salem, and it's with this recruitment strategy. And so what they're trying to do is to get rid of kind of like the drawbacks of old research where you just got a whole bunch of like 20 to 30-year-old white males. Like they just wanted to have like a way better diversity so that we can get more accurate results from what the medicine is actually doing. And so they do this partly through community outreach and education. And so they'll go to places where just normal people are, but a lot of people. So think of like the healthcare clinics, the food banks, back to school drives, and at these events, they're not trying to recruit people specifically for any clinical trial. All they want to do is educate them on how clinical trials work, why they might be interested in joining, how important it is that everybody's included in the trial so that we get better data and that more genetic diversity is represented. And so that's like their face-to-face component. They try to do about 1 a month. So that obviously this time of year is going to be higher for that face-to-face because this is when you have a lot of the back-to-school events. You can get whole families that way, so that's kind of nice. And then they do some targeted online social media, like kind of send outs where they just like will blast the social media, but they try to, with their logarithms, they still try to get like, we want this age group or this gender, so they can kind of streamline it that way, but it's not specifically to a specific person, right? They can use direct emails, but they don't kind of like throw out random, like they don't buy a list of emails and then send out invitations. In order to get a direct email, they had to have like filled out a survey online or click the button that said I'm interested, something like that, or at their doctor's office, they might have told their doctor, like, you know, the doctor will first present that this study is going on and that their clinic is one of the people that are participating. And so if they're like, yeah, I might be interested in knowing more about that, that's when it would open them up to direct emails. And that's also when it would kind of open them up to direct phone calls from Javara and Javara Associates. In their great big database where they kind of track all of their people, if this isn't the Genesis app, but it's a different one, they'll write on there, like when you first log in, you'll see which patients are assigned to you and what their status is, which, like if they were just like, oh, I'm interested in not just this study, but I'd be open to other studies too. Well, that's when they get their record of all the things that they may or may not have and any of the clinical trials that are currently going on that would match those ailments. That's when they'll get like a phone call to inform them that like, hey, this is coming up, are you still interested in it, can we meet with you, etc. And what they would do then is they would go, if the person's like, yeah, I'll be interested in that study, then they go into the clinic, they have like a screening before they start the trial, and then they either get the placebo or the real thing. Their doctor monitors them for a set period of time for different things. It's a different amount of time. Some of the like vaccine trials, sometimes they can only be for 3 months, right? And especially if it's a high-risk group, you don't want to expose high-risk groups and just let them be unprotected if they got a placebo. So, you know, you kind of got to look at those. Sometimes they're looking at long-term effects, so they might follow somebody for a year. With Javara, they try to like look at everybody, but they're still located in a lot of big metroplexes because you have to have a large enough patient base in order to get your studies pretty much covered. And they want to make sure that you have a wide variety of different patients as far as race, ethnicity, age, gender, and I wrote environment on there because definitely people in different areas of the country eat differently. Like your restaurants are not the same as Texas restaurants, probably way healthier. And this is just going to give us better overall data on the medication itself. Sorry, if you guys are seeing all of my Spanish subtitles. I have students that are English second language learners, so that just comes up automatically. So with Javara, the one thing that I noticed is you don't have to have any medical background or science background whatsoever. Most of the people that I met actually are not from a science background or medical background. Of course, we do have a doctor that oversees all of the studies, and then you do have nurses and doctors at the clinics. But the actual workings of Javara is kind of almost better served from somebody who's like business oriented. They're really good with organization. They have people skills. They still want to talk to people. They've got some like self-discipline and computer skills because you'll be online a lot. You know, so you have to like be able to, you know, set your timeline and meet those deadlines. You do meet with a lot of people, and they are very punctual with their meetings. So it is like, if it starts at this time, it starts at that time, and it ends exactly when it ends. So they get it down to the minute there. That's because most of the jobs are done remotely, but not all. So like the patient recruitment and outreach, part of that's online, part of that's in person, right? But obviously the clinics are in person for the most part, but not all, because some of that follow-up can be from the nurse or the doctor calling, it's usually the nurse. And just like getting an overview of like, how are you feeling today? Have you noticed anything weird? Do you have a headache, whatever? And so they'll do that part online or over the phone. And then they have a person from each region that oversees what each clinic is doing. Those are the people that I worked with over the summer. And they really have to keep the other people interested and motivated, because the doctors' clinics, they don't need Javard to stay open, right? They already have their own business. And they're just kind of participating in these clinical studies, like 1 or 2 of them a year on the side. So sometimes it's hard to keep their staff and punctual. If you need a feedback from your patient every week, then, and your clinic gets busy because maybe it's like the middle of flu season or something, it can be hard. And then I noticed that some states are harder to work with than others, just because of the nature of the things that Javara is responsible for testing. Like if they're testing new birth control devices or medications, and you're in a state where abortion is no longer legal, are you going to get enough patients from that state to represent? Because they don't have a plan B. So, so there's just things that maybe I didn't think about before that I would think about now. I know you can see my screen, but now I'm covered over my stuff. Okay, so the biggest thing is companies like Javara, they help the FDA they help the companies receive FDA approval faster, because they're the middle person. The company doesn't have to mess with the clinics, and Javara is dealing with the same population of clinics every year. So it's easy for them to be like, oh, hey, it's flu season, we're going to be testing the new combination vaccine, so we got to get up and running. I mean, they can pre-plan. Where, if you were just doing it straight from the pharmaceutical company, you would just need more people on the ground, and it would, I don't know if it would really serve it. I think that their money is better spent having a company like Javara to do the middle ground. So at any one time, Javara has about 30 clinical trials running, and their clinical trials, they involve stuff that just like your primary care physician is going to be able to handle, but they don't do specialty stuff. Like they do some cardiac things that you might have a specialist for your cardiac, but they don't do oncology. They don't do different rare diseases. There's a lot of things that they don't do, which is good, because then they can specialize in the area that best suits them, and another company can take on the other types. So the government only allows you to reward participants, like patient participants, with so much monetary funds. Like you can't say we paid you to give your opinion, right? So they work with organizations such as the Greater Gift, and the Greater Gift can provide incentives to potential patients and patients that kind of maximizes what they get in return for participating in the trial. So the trial might be able to give, say, $50 a person, so they might get a $50 Visa card or something like that when they go in. But with the Greater Gift, they can provide they can provide them a dinner, or just for listening or participating, or gift cards, or just things like that that kind of helps sweeten the deal, kind of helps get people interested, and it doesn't cross that federal regulation of how much money you can actually give to a potential participant in your study. And I think that's it. That's all that I have for that. So I can unshare my screen if I'm not. Overall, it was a pretty good experience. There were some things maybe I would have done different, but overall, it's a really vital, important part of the industry if you want to get your pharmaceuticals out there quicker. The other thing that Javara doesn't work with is they don't work with veterinary medicine. And I know that a lot of times you can get something approved through veterinary care before you get it approved through human care, but they don't work with that part at all.
Thank you, Yvette. I'm going to ask if there's any questions at the table. We'll repeat the questions because you won't be able to hear them if we don't. So, Any questions? Okay,
How is she?
Rafi,
Hi,
come on up. Don't have to repeat this one, she's here.
How are you gonna share that with your people?
There you go.
So at my school, it's an early college high school, so I get like two types of students, right? I get the ones that, they definitely want to, obtain like a PhD or an MD, and they're gonna go a different route. But for the kids that they've had med-turn, they've maybe had some farm classes, so they have a little bit of the medical science, but maybe they don't want to jump in and work with patients. Maybe they're not somebody who's like a hands-on type of person. So this is the students that I'll share this with. I've actually already shared it with some who have recently graduated, because it is mostly a remote job. Not all, but mostly. It's something that they can do and still have a family, or still take some classes but work at a different time of day. As long as you make your needs, you can work whenever you want. So I'm going to share it to them in that way. If they want to go into bioinformatics, I would tell them to go more towards the pharmacy. Bioinformatics is one of the new degrees that they can get from right down the street. So if the kids are going to stay local and not go into research but still want to use some of that knowledge, that's one of the things that they do. But I think that this would be more for the person who is more of a hands-off person. Perhaps they don't really do, if you go into medicine, you have to touch people. It's just the way it is. But you can still be in medicine with this without having to have that interaction. So there's definitely a group of students I can definitely share this information with. And how I would do it, I would just do it in the clinical studies unit that we already do. When we talk about the different types of clinical studies, then we'll just add this to our career portfolio of what possible careers you have in this field.
Very good. Rafi says, good job on answering the question. Any other questions? So I'll make a comment. I've been in 2 of the trials that Javira offered. I think you described the process perfectly. As a young man, I used to work in the industry, and I used to design clinical trials. And Yvette, the one thing I noticed when I was in my trials, and I brought this to the attention of the principal investigator for the Javira trials, there's still a lot of paper being used. Did you see any paper being used?
No.
Okay.
Okay, everything is literally online, but so if you use Excel, then this is your fine. Or if you know how to research, because one of the things that was kind of, I don't know, there was not a really awesome database to find out your stats on all kinds of different diseases for every area. So the CDC does have one that breaks it down from state and counties. And I guess when I do research papers, I do it a little bit differently than when I'm presenting our meeting, because we had a young intern, and she's way faster on the computer. She was great at Excel, But I would prefer if everybody got their data from one source and it was sourced. You know what I'm saying? But there isn't a lot of paper. There's a lot of paperwork, but not physical paper. Maybe in the clinical area, they do have a questionnaire that they fill out, but I think most of them are using iPads now.
Good. That's far better than during the COVID period when I went on 2 trials, and then the second trial, first trial was COVID, the second trial was a flu vaccine. And I was talking to the PI about a month ago, and I said I was just flabbergasted that they were still using paper, but he said, that's not them, it's the sponsor. Right? Okay. So,
yeah, I think that has to do with the individual sponsor.
Yeah. Thanks, Yvette. Really appreciate it.
Thank you. Do I need to come back later and talk to Ms. Amy?
You can if you want to. We're going to be eating.
Okay. Okay. Working lunch.
Yeah.
Okay.
We do have a certificate of precipitation for you. We're going to send it to you. Okay.
All right,
participate.
that sounds great.
Yeah, thanks, Suzette. Great to see you.
Thank you guys. Have a great summer. Bye.
Thank you, bye.
Our next speaker is Pratima Bashar, and Pratima, you're going to tell everybody that you're actually in here from where?
From Bangalore, India,
Yes.
like 8,000 miles away from here.
So you worked at W Firm for the summer? Right, so come on up.
Hi everyone, this is Pratima Kocher here. So I'm here to present my summary for the NSF ATE Beta Skills Summer Fellow Industry Program. Okay, so I worked in Carolina University and I'm also associated with FTCC. A little bit introduction and background. I worked as an assistant professor of biology and program director of biology and public health in Carolina University, Winston-Salem. And then I did my PhD from Tohoku University, Sendai, Japan in biomolecular sciences. My master's is from India in biotechnology and my bachelor's is in industrial microbiology from India. So I was placed in W Firm, which is Wake Forest Institute of Regenerative Medicine, and they have a department called Translational Core. So what does Translational Core does? They just scale up the research going on in W Firm and and put it into products, manufacturing. So translates bench research into manufacturing products. They do not do functional assays, they are more into manufacturing, but they do perform regulatory and functional research. What does that mean? They do the endotoxicity, they test for tumor cells, and then tests needed to scale up the products. So main goal for the Translation Core is to manufacture any product. It's mostly they are producing cells. So they are working on the TC. They are working on like 10 different projects. Name a few is erythral tissue engineering, hemophilia. In hemophilia, they are producing cells. So they're genetically engineering cells which can produce the factor VIII, which is useful in blood clotting, so that they can inject those cells into the patients. They're also doing work on bladder reconstruction, engineered penile tissue, engineered vaginal tissue, and amnion hormone. And there were many more. There's just a few of them here. So to start with the trainings, we did all of us, me and Rafi, we did the initial trainings, which is for everyone who is joining the W Firm. So it's GDP, good documentation practices. We did it with Gillian Moore. So we learned about how accurate, clear, and consistent documents is needed in the manufacturing department or companies. And then we had a cell culture training with Lab Tech Steven. We mostly talk about how to maintain that core lab and how to dispose waste. For histology, we had training with Cindy Zimmerman. There were like different kind of trainings we did. We learned about fixation, how to fix the samples, then how to stain the samples, how we can embed them, and then cryosectioning, like how we can make the section or the slides for the IHC stain, for the staining. And then there was last training was imaging. So it wasn't just an overview of what kind of microscopes they have. They have confocal microscope, fluorescent microscope, they have SEM, scanning electron microscope. So it was an overview training, but if someone is ready to use any kind of microscope, they're ready to give a specified training to them. So particular project I worked on, or I would say I shadowed the people who were doing experiments. So I was a part of two main projects, hemophilia and EPT project. In hemophilia, it's a cell therapy, which is so what they're doing, they're from donor placental cell, they're taking out the cells, they're engineering those cells so that it can make more factor VIII. So the levels would be sufficient in the patient. So for that project, I shadowed the experiments of biodistribution of human cells. So they have the product, they have the cells which are producing more factor VIII, they put it in the mouse, and we were checking how they are distributing in the mouse. So how many organs, which organs, and we use staining to test that. So IHC staining to test, which has a human-specific antibody, and then we did the qPCR to determine the standard curve, like how many cells are there and how they're surviving there in the mouse cells. And these are human cells we're talking about. And these human cells are the product they already made. For EBD project, I mostly shadow the experiments which involve the IHC staining. IHC staining is most, used for the destructive testing of the seeded construct. So seeded construct is they have a scaffold, they put their cells on, and then they proliferate. And then they have a product and they can put it back into the patients. Apart from the research or the testing part, on the management side, I learned how the project flow in that department. So basically, the PI would have the project, it will go to the translational core, and then they have meetings, trainings, how to make that product. And when it is decided that particular project will go to the quality assurance, they properly document it, they will write the SOP, everything is documented, and then when the procedure is documented, it will go to the quality control, they do the testing of the product for its safety, sterilization, endotoxicity, and watching if there are any tumors developing or not. So all this testing is being done in the translation code. And then when every testing has been done, they provide the documentation for the IND. When it's approved, it goes to FDA. FDA has the 30 days to approve it, and then goes to the phase 1 clinical trial. So WFORM is involved in phase 1 and phase 2 clinical trials for many of the projects. And products they have already in the patients. It's EPT, they did change a piece of fibrotic and necrotic pieces. And for bladder, phase 1 is already done. They are preparing for the phase 2, EBT. They already have 2 patients, and they are in phase 2. And I think this next month, they're treating the 3rd patient. And MDM powder, they're using that powder for wound healing, in diabetic patients. Phase 1 is done, and phase 2 is in process. Other things I learned about this department is the quality assurance department and quality control department. They're the pillars of this core. They work together, and what are their duties for the quality assurance? They basically work on the documentation. So they maintain, control, train, audit services to the surgical core. They do also maintain the website, quality assurance, auditing, document control, control of quality system, and many more. So mostly they are working on the website and the documentation. Quality control, they are working on testing as well as documenting that testing for the product. So taking samples of the product, creating test solutions, running tests of products, and then documenting and analyzing test results. And apart from WFORM, I would like to thank Dr. Aaron for arranging a tour for the Novex Innovation, and special thanks to Ryan to give us a tour. It was very informative. Thank you so much. So in that tour, we learned about how actually this industry, this company runs. So clean room operations, how they prepare media and sterilization. They have different departments for packaging, for distribution, how they process the human tissue, and qualification in industry standards. We talk with Ryan about what kind of skills you are looking for the students who wants to get into the industry, and it was informative. So as in summary, they do provide an insight how this industry operates. So that's an additional learning. So for this slide, I specifically want to thank Ryan, Dr. Stephen Burr, Lisa of the Department of the WFORM team, and there's one more. So we have discussions. So I have like individual discussions and I have meetings with people, and specifically I want to ask them a question: what skills you are looking in the students to hire them? So I summarize a few. So they are looking for the students who have a little bit of exposure of GDP, GLP, cell culture technique, aseptic techniques, SOPs. They should be team player. They know how to follow the instructions and attention to details. So soft skills are also part of that list they are looking for. They will definitely give a specific training to every person coming in. So what I have learned from here, or what I can do to implement this learning, if anyone is integrating a course in their program, then there should be a biomanufacturing, like if there is a biomanufacturing course, you should expose your students with the terminology. They literally have their own language. For first week, I could not understand what they're talking in the meeting. I mean, I couldn't. So they do have their language, so students should be exposed to that language, that terminology. They should know, even on documents, at least some exposure that what is SOP, what is GDP, GLP, GMP, I'm still learning. I had no idea. So, yes, and if someone is planning, or if I'm planning to create a biomanufacturing program, addition to all those terminology and all that concepts, there must be a training in the industry, at least for a semester. It is needed. Because anyone I'm talking to about the skills requirement and what the advice you have for the students who want to join the industry, they all are saying that you will learn when you do it. So it's very essential for them to have skills-based learning if they are in the industry. It's beneficial for the students. And then we also have a little bit fun time. So for the first picture, you can see my colleagues, Evet and Rafi. We went to, there was an event in the Winston-Salem downtown, and Aaron, Rafi, and me went to hiking, right? Yeah. And then we went to visit Duke and Chapel Hill University. There you can see us having lunch. And that's a picture from the Duke, and then we attended the Bio Night, Tribe Bio Night, Jeffrey, Aaron, Evet, and Rafi. And in last, I would like to thank you, Russ, for this opportunity. I didn't mention many names, so I'm going to add it now. And it's a grant I would like to thank. And I would like to thank Dr. Steve Barr to take me into the translation core. And then Dr. Vladimir and Dr. John, they were the PI of the translation core. They were doing the research needed for the core. Dr. Bida, she explained very well how the project development and the process development happens in the translation core. The whole TC team, so this is the translation core team. They have many people working in the QA department, QC department, and they are lab techs. Dr. Aaron and Ryan for the tour and arranging the tour. Thanks to Zach and Sach for allowing me to get into their lab. And a special thank, I didn't mention, to Rafi, whenever I feel blue. It's a special thanks because she was there whenever I feel blue. I'm far away from my home, so thank you. And that's it for my presentation. Thank you.
Nicely done, Akila. Thank you. Any questions?
So you were mentioning with WFirm the project workflow, and so after phase one, if there's a good indication, is the intent to then spin out a company from WFirm, like create a company specific to that product?
You're going to have to come back here and just repeat the question.
So I
So if there's a good indication with phase 1 studies, is there an intent to spin out a company from W firm to create that product?
So so the question is, if the clinical phase 1 studies are a good indication, they are moving forward for the,
Yeah, like a separate company
For making
being generated from W firm at that point.
they might have some collaboration, I'm not sure about that, if they are like,
But Dr. Green might be able to answer
Yeah.
that a little bit better.
Sorry, I didn't.
It's whether or not they're going to take the
they're taking in another
and
companies to make
spin it
the
out,
products.
spin it out as a company. Which one are the?
Yeah, so I was just wondering if the W firm workflow.
The workflow.
So, like, from the translation forward, once we get into phase 1, if there's a good indication, you can spin out a separate.
Yeah,
company.
in general, go through phase 1 internally with the translation board, the phase 2.
Mhm.
We have capability that every firm has the capability for phase 2. And then if you have success through phase 2 trials, then it's either spun out into a company licensed, perhaps the technology or science, then it's licensed to someone else through a collaborative agreement or, but the phase 3 trials can all get done externally.
Okay.
And that's one of the things that I came to this sort of as a lay person, I didn't know a lot about it. One of the things that I've learned is, and having experienced some other research centers, not unlike W firm, not as good, but not unlike, is that W firm really does take potential products and technology further in the clinical research trial area, in the translation process overall, than you find in a lot of research centers in general medicine. So once something comes through W firm, it gets to the point where it becomes sort of handed off to some type of commercialization process.
Okay.
It's further along sometimes than what occurs in some research centers.
Great. Thank you.
So the question was, if everything goes well with the phase 1 clinical trial, did W firm spun out to other companies for producing the product? And Dr. Green did reply that W firm has the capability of doing the phase 2 trials, but after the phase 2 trial, if there is a need, there are some cases that they did reach out to other companies to make the products.
Any other questions?
I have a question to the science, since this is originating cells from placenta.
Yes.
Is the product per se from pooled from the number of people, or is it customized?
It's actually it's unique. Every product is from the patient. But for placenta, they do have, like for amnion powder, it's a bag, the placental stem cells bag. They're making amnion powder from the bag, so it's a pool. So it depends on the product.
Depends on the product.
Yes.
I have one follow-up question. One of them you're talking about with they were producing factor 8.
Yes.
Is that, are the cells naturally producing
Yes.
that?
Yes.
Or
Their cells naturally producing that.
okay, wonderful.
And the target patients are the parents who both have hemophilia.
Thank you.
Any other questions for Patty before she goes back to India? Radhima, I want to thank you very much for your excellent presentation, and thank you for the people that assisted you at W firm.
Thank you so much. Thank you so much.
Thank you very much. Well, that concludes our presentations, and I want to thank everybody. I want to thank the fellows, and I want to thank Olivia, the intern, and I want to thank all the mentors, and so Ryan, Dr. Green, Zach. There's many others out there that aren't here today, but it's been fantastic, and I love, having come from industry myself personally and spent a long time in industry. It really in my travels, and I used to tell this to Dr. Green all the time when he was at the college, that I noticed that the people that I interacted with, I could tell the people who were in industry and the people who weren't. It was very clear to me. And so I always had it in the back of my mind that this program would be, a great program and a very successful program, and I think it has been and continues to be. So thanks very much.